Hello, my name is David and this is V, and we are here to go over the theories of DNA replication and how DNA replication works. First, let's start off with theory and discuss the three models that were proposed for DNA replication. There's the conservative model, semi-conservative, and dispersive. The conservative model involves the parent DNA being copied, leaving us with the original parent DNA and an identical daughter DNA molecule that is comprised of two brand new helices. The second model is the semi-conservative model, and it states that the helices of the parent DNA are separated and each helix is complemented with new corresponding base pairs. In the end, the daughter DNA is made up of one helix from its parent DNA and one helix of new DNA molecules. The last model is the dispersive model. This model proposes that daughter DNA is created through random interspersion of parental and new DNA segments. Of all these theories, only the semi-conservative model was proven to be correct. DNA does in fact duplicate by adding fresh DNA base pairs to the split helices of parent DNA. Now we will talk about Meselson and Stahl and the experiment they ran to figure that the semi-conservative model was correct. They grew E. coli in a lab and gave the bacteria DNA made up of isotope 15N. This was to distinguish the parent DNA from the new DNA that was made up of isotope 14N. The DNA sample was taken to a centrifuge to be separated so they could establish the makeup of DNA in the duplicated molecules. DNA molecules containing isotope 15N are considered heavy, and molecules containing isotope 14N are light. A combination of the two is called a hybrid. After the DNA samples were taken out from the centrifuge, heavy DNA sunk to the bottom of the test tube, hybrids took the middle, and light DNA was at the top. As you can see in the diagram, the samples are separated by generation, and the only model that fits the pattern is the semi-conservative one. This concludes the theory segment of the lesson. We will now move on to how DNA replication works.
Now let's take a closer look at the semi-conservative model. DNA synthesis or replication is pretty intimidating, but we can break it down into 4 steps: initiation and unzipping, priming, elongation, and proofreading. The ORI site is where our adventure begins. In order to duplicate our DNA, we're going to have to unwind or open up our double helix. This is when our first two enzymes come into play. DNA helicase will start at the ORI site and unzip the DNA in both directions by breaking the hydrogen bonds between the base pairs. DNA gyrase helps by uncoiling the DNA. Together, helicase and gyrase continuously open up the DNA. Once the DNA opens, due to the nature of the base pairs, the DNA will want to recoil on itself, so we have single-stranded binding proteins to make sure once the DNA is open, it stays open. At this stage, we have now what's called a replication bubble. For simplicity's sake, we like to cut this bubble in half and look at what's called a replication fork. However, we should keep in mind that the exact same processes are occurring on the opposite side. Before we can build our DNA building enzymes need to know where to start. That's why we have primers. Introducing DNA or RNA primase. DNA primase places down RNA on our strands to act as our markers.
Elongation. Once the DNA primase is in place, DNA polymerase III creates new strands from both parental template strands. It is important to note that polymerase III will always attach to the 3' side on the template strand and run in a 5' to 3' fashion, and that the two template strands run in opposite directions. This is key because it tells us that DNA polymerase III will also form new strands in opposite directions. The polymerase III that is working in the direction of the replication fork is able to lay down new base pairs without interruption because the replication fork and the polymerase III are moving in the same direction. This is called the leading strand. On the other hand, the polymerase that runs opposite to the replication fork is called the lagging strand. The lagging strand adds new DNA in segments rather than in one continuous line. These segments are called Okazaki fragments. As the replication fork opens and space is created on the parental template strand, DNA primase is laid down so polymerase III can get to work. This process is repeated until both parental template strands are completely unwinded and the new DNA has been placed down. Then a couple of new enzymes are introduced, the first being DNA polymerase I. Its job is to remove DNA primase and replace it with new DNA nucleotides. Then DNA ligase comes in to act as an adhesive for the new DNA strand, binding the Okazaki fragments together to form a single strand of new DNA. Now we've got a fresh strand of DNA, but wait, we have one more step.
Proofreading. You wouldn't submit an essay without at least reading it over again, and that's what happens in our DNA as well. Polymerase 1 and 3 go over our newly formed strands and check to make sure all the base pairs match. We can tell a pair is mismatched because they happen to form a lump. When a mismatch is found, polymerase 1 and 3 will cut the strand and replace it with the correct piece. Finally, ligase will seal any gaps in the new strand. Now you know how DNA replication works and the theory behind it. We hope you learned something today. Thank you for listening. And also thank you Vishnu for his moral support.